bit of coffee. I was asked to make a reminder that. What's that thing? I was asked to make a reminder that there's no smoking on the campus, and that security may ask you to leave if you do. Oh, I see. Thanks, Dana. So my name is Alan Yarmish. I come from the National Institute of Environmental Health Sciences, so it's part of the National Institutes of Health in the US. And I have the pleasure of being here today to introduce to you a little bit about the data set that we'll be working on through the workshop, and carry you through a little bit of the initial steps in terms of getting access to the data, and giving you perspective in terms of where this data came from, the objectives of the workshop, and in terms of the exercise that you'll be running through. So a couple initial tasks. It may take us a little bit of time. We ran into some permissions issues. We're going to initially first try to get access to the data. So this is data that was published in PLOS ONE, collected by my mentee Morgan, who's lead author on this paper. So you can kind of see our analysis and the results and compare it to like what we get out of the workshop. This data is accessible through Massive. So this is one of the Metabolomics data repositories that can be used. And the Ascension number is listed here, Massive 29307. So Daniel is going to help us because this computer has permission issues. So there's a couple different ways you can access the data. One is to do like an FTP kind of link. Let's see, Daniel, perfect. So the easiest way is to navigate to massive.ucsd.edu. And if you have issues, some of the instructors can help. And then if you go down to the browse publicly available data, there's the left, and hopefully you don't get a server error. Let's hope Massive can go down. All right, one more time. There we go. So if you search for in the title box, there's a little search box right there. (29307). Nine two. Yeah. Remember, you can blame Daniel for searching the wrong thing. There it is. Summer school. So if you click on that green box, green highlighted text, this will be, so this description, so every public massive data set has a little bit of a description to it, some contact information. So like I'm listed as the PI, and then basic information about the file sizes. Most importantly, right where Daniel's hovering, there's the FTP download link. So if you copy that address and then paste that into, if you're a Windows system, you can use the File Explorer. And if you just paste that in the search bar, that's one way to access it. So that may not work for everyone. So if you have problems connecting, let us know. Can everyone get there? A username and password. Okay. So there's an alternative route that can work. So we'll try that maybe. But yeah, I heard a couple issues. But is it working for others? For you it is. You have a username issue? Okay. Okay. So to our multiple people being asked for a username, password, okay. There may be if you already have an account that's linked to your computer, this file directory thing may work directly. If you have not signed into GMPS or Massive before, it may be asking you for a username. So there's always the option of signing in to Direct Massive, and that will allow you to get to this. The other option that we'll go through doesn't require you to have a username at all. Okay, yeah, so that may be slightly different. So. So not the massive one. So for those of you who are having permission issues through that mechanism, Daniel's gonna walk through the alternative. So there is a way to use like a file transfer protocol software. So FileZilla is no longer suggested, but it is possible to use. But WinSCP, I believe, is the one that's recommended for GMPS right now. So if you have that software on your computer, you can open that. So FileZilla works on Mac. Yeah, so that one, we're gonna get around the username. Just name not. So those of you who are using WinSCP, you'll be greeted with a login screen. And it should default to the first option. You want to go to the drop down, select FTP, and then do no encryption. The host name is massive, massive.ucsd.edu. And the port number is 21.
We connected the Zoom.
Oh.
Yeah.
Okay. Yeah, sorry for those on Zoom. So the FTP, no encryption, massive.ucsd.edu, port 21. And then for the username, you want to put the massive number. Fortunately, it has to be the entire thing. So it's MSV 4092307. And the password is just a lowercase.
If you have problems seeing the screen, the resolution, just let us know.
Devices that people can disconnect from their phones. There's some people that hate that.
So I was just reminded that with the wireless password, it only allows you to have one device on the network, so there is limitations. So if you have your cell phone and computer, it may kick people off of the main network. So it's advised to only connect with your laptop computer.
The actual for the dataset. For the MSV. Yep.
Oh, okay.
Try.
So
Yours is not where. Go.
92389. Oh, it won't.
Oh, you have something in the password.
It's this computer won't work anyway.
Huh.
So
Better tell the people that they have the correct.
yeah,
You have it?
I was gonna just type it. Slight update for everyone. I was told that we actually re-uploaded the data because there were some incorrect files. So the real number is sorry, this is the hands-on bit. The real number is 92389.
It's a lower. Yeah.
Also, what I'm going to do something, there's like a thing that you can also
I don't know. No, it doesn't come. Ah, wait, it's like 4 zeros. No, not necessarily. Are you sure it's the lowercase? Can you get it on the website? Yeah, we all have the same problem. Yeah.
The website's not loading.
my Wi-Fi is just going down.
So I talked to 5 people. We had 5 different issues. So we'll try to address these one at a time. So bear with us. So handling, transferring data can be challenging in metabolomics. So did it work for anyone? Okay, 3. Okay. So we're,
That's what the day, the case base.
Is your Wi-Fi working?
Yeah.
I know, but I try and get.
Found one guy, I'll make sure
My,
you translate it.
yeah.
You don't get it, your wife is the only one down. I'm not gonna use my.
Yeah.
I'm sure you want to.
Yeah, and I just tried to reconnect with it.
Okay.
I think it's the Wi-Fi.
so I think we've resolved the main problem. Or at least one of the problems. Multiple people losing Wi-Fi connection. Someone hotspotted and was able to immediately do it. So we don't actually need to access the data right now. So fortunately, so we have some time to figure it out, but we will ideally get the connections figured out, because we'll need that in subsequent days. At the very worst, we'll bring it on a flash drive or USB, or you can download it at your hotel. So apologies for that. Some of this may be Wi-Fi permissions. It was noted from someone here at SSI. Maybe this looks like a denial of service attack, right? With all these people accessing it at the same time. So which is quite possible. So apologies for that. But there, and we'll update this slide as well, and share it with you, but the data, it should be easy to access through Massive. And there is documentation on Massive as to how to connect through different mechanisms and actually obtain the data and download it. But in principle, all the data is there, it's accessible, you can download it. And then some of this information is also on the GitHub repository that was mentioned multiple times. And if you search for the Metabolomics Summer School, I believe that it's the first hit that pops up. But as we move past this, I want to give a little bit of perspective to like what this data is, where it came from, and what generally the exercises we'll be walking through are aimed at actually uncovering. This is a little bit of a, so if you bear with me, a little bit of a story, how this particularly became a research interest of mine through my postdoc years, and kind of has continued in different ways in my current position. But before I even joined the postdoc group, which is Peter Dornstein, you'll see a couple overlaps here from the instructors. So this publication came out, apologies for the small text, but it's in PNAS in 2015. And I'm gonna go through a little bit of these. But to summarize it at the general level, this is kind of how I kind of wrap everything up in my own brains in terms of the process of discovery. So from that paper, there's some really interesting observations, but there was not the ability to actually support it with other information, right? But it led us to develop these hypotheses of what actually might be going on. So then we designed, you may call this a pilot study or something like that, right? Where we actually still found interesting observations, but because we had designed the study to be open-ended to discovery, but we collected a bunch of additional information about these particular subjects in this case, we were able to actually corroborate some of the observations that we found. So instead of just being like an anecdotal, one time we saw this, but we don't know if this applies to multiple people. Now we have a bigger cohort of people where we can still make some interesting observations, do discovery, and have some information, but maybe not directly tie it to any particular mechanism. And then go through and actually do modeling and prediction So this is all about looking at like what chemicals can be detected on the skin.
That's what you're showing this one. Thank you.
Oh, perfect. So moving towards prediction and making predictions from these observed hypotheses, and then in the end performing some kind of clinical study or validation, right? So we're going to kind of go through each one of those components very briefly, again, just to so that you understand where this end data set originally came from and kind of see how it fits in the larger context of kind of doing a metabolomics study. So this initial paper that I mentioned, so this is again in PNAS. So this was the molecular photography of the human skin. So what they ended up doing is actually taking two subjects, one male, one female, and swabbing like tons and tons of locations on their body, right? And then looking and then doing LCMS analysis, so metabolomics, on all those individual samples, and then looking at the distribution of individual signals and how they occur. So I saw this, I was super interested, and we'll talk a lot about molecular networking and some of the tools that were used to analyze this data. But I saw this particular set, so this is sunscreens, and unsurprisingly, they were found on the skin, right? So avobenzone is one example, and in this particular individual is found around the face and neck, right? So kind of where you would imagine applying sunscreen. But it kind of got me thinking, so this is interesting, right? So this is kind of an interesting observation without any ability to kind of corroborate or generalize this to a general discovery. So we set about actually doing a discovery pilot, right? So the intent of all this was to see if we could use skin samples as a way to non-invasively monitor drugs that people are taking, and so what we set out to do was, this was a cohort of individuals who had kidney transplants, and for numerous reasons, and this population of individuals is on a lot of pharmaceutical drugs to prevent transplant, prevent infection, right, and to maintain good health. So we thought this was a good population to kind of take a first stab at to see if we could actually see some of these drugs on the skin as opposed to the sunscreens and things like that. So the way we did this was we took a bunch of skin samples, again, similar kind of methodology, and basically just swiping the outside of the skin. So very moderate pressure, but just like a wet swab. And then in this particular case, we did multiple places on the face, the armpits, the axilla, and the hands. And that was kind of chosen on purpose, but also, it's again, discovery. We're kind of doing something new, so we didn't really know where to swab or what to swab with. So we kind of went through and did this laborious process. And I'm blanking on how many individuals, but I'm guessing there's 15, given that there's 15 little stick figures there. But we had multiple time points. So these individuals, because they're on transplant regimen, they're continuously monitored. So we have pretty good medical history for these people. It's a little bit complicated in the United States in terms of medical history. So we don't have electronic medical records. But we were able to basically track like what people were prescribed, how their prescriptions changed from one visit to another. I think the average duration is something like a month, 4 weeks in between the two visits. And then kind of similar as we've seen before. So we used a QTOF type of instrument. We did LCMS, and we basically just did a simple extraction, cut the swabs into a tube, and then extracted it with solvent. Skipping all the details of how that study was done. But if you have any specific questions, we can go over that. But this is displaying some of the results. So timolol is one of the drugs. So that's the top two pictures, A and B, was prescribed to only one individual, and only in the individual it was prescribed was it detected. Interestingly, it was only found in the first visit. So that would be this panel right here. You can see my arrow. And as the second visit, they dropped. So red is essentially detected. So this is a fairly binary, non-quantitative assessment. A similar story with citalopram. So this is another drug that was prescribed to someone, one individual, and in this case, it was prescribed on both, and taken on both. Same thing with diphenhydramine. So this is a antihistamine, very common, very old drug. And this will come up in our data set as well. And then N-acetyl sulfamethoxazole. So this is a metabolite of a antibiotic that is prescribed to these set of individuals prophylactically, so to prevent infection. The general observation of the study, again, these are sometimes only 1 or 2 people, so hard to generalize to a general kind of population. But because we have prescription history, we had multiple visits, and also we then could tie the people who were taking sulfamethoxazole were the only ones that actually were detecting the metabolite in the armpit. And the question is, why do we detect it in certain locations? So like there's probably different mechanisms. So, and the acetyl sulfamethoxazole is much more polar than the initial drugs. So the idea is that maybe it's coming out in the sweat of the armpit. Whereas the parent drug, diphenhydramine and citalopram, were found primarily on the surface of the skin. And the hands were always kind of suspicious because you're worried about passive contamination. So if you're opening a pill bottle and opening it, just that alone could maybe transfer enough that we could detect it. But it was harder to explain why you saw it on people's face and through multiple time points. So this kind of got us to thinking, well, maybe instead of just guessing at which drugs and which people, maybe we should try to predict it. Oh, but this paper came up in the middle of this, which makes the connection back to sunscreens. So this is sunscreens not coming from, so sunscreens applied to the skin, right? Makes sense that they're there. But this paper came out, so this is a study from the US Food and Drug Administration, the FDA. And here they showed that if you actually apply sunscreens to your skin, it's then detectable in your blood. So it's actually going from skin topical exposure into your systemic blood. So the idea was kind of reinforced. Well, maybe there's things that we take orally, go to your systemic blood, and then become available to the skin the other way. So it kind of gave us hope that this kind of line of inquiry was going to pan out. So this, with a lot of help from Wout, we developed basically a model to predict which chemicals or which drugs actually would be available on the skin. And we basically took all the structures of the drugs in the drug data bank, drug bank, I think is the database, but it has all the listed structures and identifiers. And we predicted a bunch of different chemical properties like log P, the size of it, the polar surface area of the molecule. And with that, I mean, it's not great, but 0.7, was, our area under the curve in terms of predictive ability. So then we started retroactively applying that to the drugs of our choice. And I can scoop this over. So this is citalopram. So this is one output kind of showing the model. So the more red essentially it is, the more likely it was to be found on the skin through the model, or predicted to be. And the more blue, it's the less likely. So citalopram is one of those drugs that we saw on people's face surface. So again, it's not a very high prediction, but it has a higher likelihood of being found in the skin. Contrast this against tacrolimus, which is a drug that everyone in that cohort was actually prescribed as an anti-rejection drug. And that essentially is one of the lowest predicted drugs that we actually found. And we never observed that on the skin. So kind of give us some contrast in that we might be able to detect something. And then we went about and, did the other one. So diphenhydramine was another one of those drugs that was found on the skin. And it was moderately predicted, right? But it's still predicted to be possible to detect it on the skin. And this last one, this diphenhydramine and hexose, we may talk about that later, but it's very low in terms of its prediction to be on the skin. So kind of the initial observation was like, hey, you can see sunscreen on people's skin, to go to then the pilot clinical study of like, hey, we can explore what drugs might be possible to be on skin, have prescription history, link it there. But again, that's like N equals 1 or 2 individuals. Try to predict something, and then we kind of move to this data set, which is like a specific clinical study designed to address one very specific question. The research paper title kind of makes it more open-ended. But the principal question is, can you see diphenhydramine? And that's kind of the whole premise of the study. So the way that this study was actually done is that we enrolled healthy individuals. They were 15 to 25 years of age, so around the university, right? They were paid for their participation and volunteered to do so. But we asked them to come to the clinical research unit that was on the medical campus that's adjacent to UCSD. And they sat in the clinical research unit for 14 hours. So they came early in the morning, and we provided them with food, Netflix, and $200 for their voluntary participation. So we had quite a number of volunteers, and we asked them, and we administered a single oral dose of diphenhydramine. Again, that's Benadryl, if you're familiar with the brand name. So a typical normal adult dose. Told them, warned them of the side effects that you may become drowsy, sleepy. And so they were continuously monitored through that. But the whole time they were in the clinical research unit, they were taking all kinds of, vital measurements, temperature, blood pressure, doing basic tasks. But we also collected both blood and urine and skin samples concurrently. So at each one of these time points that we'll have in this data set, we have both blood and skin actually. And so then in this study, we can actually directly link basically consuming that diphenhydramine to the observation of the level of blood, then hopefully to observing it in the skin. So I don't want to reveal anything right away in terms of what we found, but we're gonna work through this together as a data set, right? As an example. But there's 3 actual skin locations. So we got a little bit smarter in terms of where we need to measure. So it'll be the forearm, the forehead for the individual, and then the upper back. So there's some indication that the upper back is a place to, again, avoiding hands because we don't want any contamination or cross-contamination touching things. And the 24-hour time point, just to give you a reference, we didn't ask them to stay there for that long, but they came back the following morning and then supplied an additional time point. So that's kind of the data set as a whole in terms of where it came from. And then the actual exercise is predicated on answering a couple of these basic questions. Is diphenhydramine or the metabolites? So this is where the untargeted bit comes in. Because the question was, what metabolites are there? Where are they distributed? Which ones? Can we discover new ones? Like, are they biotransformed on the skin? And if observed, what are the pharmacokinetics of those particular drugs, right? So like, how does the temporal kind of distribution change? And then, the benefit of doing the untargeted, we're coupling this kind of specific question with the more broader question. Are there any other things, right, that actually change with this, given that we are also giving them snacks and things like that? And do those change over time? So everyone will be working with the blood data as kind of that's kind of like the standard, right, that we're going to compare against. And then each of the groups will actually use a different subset of the skin and kind of answer those specific questions. So the instructors have kind of worked through the upper back examples. So we have that for you. And just to detail a couple of numbers, there's it's 42 total samples. It's 7 people at 6 time points. And the bloods, again, it's 42, so it's perfectly matched. So it's 88 samples in total, so fairly small. And then group 1 will go through the forehead samples, and group 2 forearm. And those 2 were kind of chosen because they are different explicitly. So if you have any questions about where things come from, or the meaning behind it, or details of the study, I'm happy to address that. I should mention the way in which we collected this data and the instrument platform. So this was, again, the untargeted metabolomics. It was liquid chromatography, mass spectrometry. We collected the data using DDA, so data dependent acquisition. The data was collected on a Q exactive, which is a type of Orbitrap. So it's all high resolution MS and MS data. Yeah, that's it. And then, yeah, and just to kind of recap, data is available there on Massive. We'll sort that out. The GitHub repository, we'll come back to that multiple times. There's notebooks that we use to process some of the data that are available there. And all this, and our interpretation was published here. Hopefully we recapitulate our findings and don't find any mistakes. But potentially discover some new things too, because that's one of the great abilities to go back to this a couple of years later. The tools are much better. Questions? Yeah, so that's it. So the question was concerning how do we like clean people's skin from products and things like that they may use in their personal care routine. So there was an inclusionary and exclusionary criteria. So like one of the, like they could not use personal care products for the 24 hours prior. I believe maybe it was 12. But then there were other things like they were told to abstain from like caffeine, from smoking, things that we know that will pick up and on the skin, and things like that. But in general, we kind of did this as a fairly uncontrolled, typical population, right? So we didn't tell them to use a certain soap or product. I think it was requested that they don't use anything prior to. People certainly did, as we found out. But I think the principal question, it was like, in a realistic setting, could you actually start to detect some of these drugs, right? In that way, it was a little bit more specific or detailed. So hopefully like those things don't interfere with diphenhydramine. But it kind of gets to the second question, like, can you get anything else out of the untargeted data? It gets a little bit more complicated. What is next? Early what? Downloading the data, yes, we could try that. Oh, okay, so the lunch break is at 12:30. Okay. Okay, so we could try to figure out the data download bit here. So
All the Wi-Fi.
Yeah.
I could see.
Yeah.
I wonder if they like.
It's heavy. They're all just-
Do that. I mean, It's definitely a couple of gigabytes.
It's manageable to software.
No. Okay. I hope they brought enough USB sticks in. Yeah. You haven't.
Connection here.
Let's see.
What's weird? So, computers, we don't have enough to- Maybe maybe it was very cool. We just like to work, get it working.
Restart.
Yeah.
Some reason they also forget the. Yeah.
No, still not happening. Oh, no, I might be going in.
36 gigs. 36. Yeah. It's not bad.
Thanks.
Yeah. I have a USB hard drive.
Yeah, I don't.
Yeah, me too. When it's done, we. Maybe guys good. Trying to register the original. No, like,
I have to imagine all the people automatically integrate. Yeah. Oh yeah. Easy peasy lemon squeezing. Although.
But yeah, make sure you just have note of all of the logins, so you have to do it. Process. Down the data, you will check it. Because again, the most important thing is how guys. Yes, good luck on the download. What? The data's 20 gigabytes?
I'm actually going to do that in the personal.
Don't mess with my law.
I think that would be.
So I think the file, folder slide, they're converted files. It should be the raw data files. So that might help your download. Yeah, sorry.
I don't think this one. No.
It's not that. Yeah, you're okay.
Okay, maybe wire connection. There is a wire connection.
Yeah, we have, yeah. You know, already. Yes.
So maybe just say what we can have.
Because that's gonna be like reducing from like 20 gigs to 3 gigs.
So, yeah.
Yeah.
So can you, I think you can see it from here,
Yeah.
Yeah,
right?
should be possible.
So at least there's like
We need,
the
Peak. Yeah, the peak and not the
we
CCM.
need these, right?
Yeah, and then there's example and
Yeah,
then
there's
group 1 and group 2, I think.
Yeah, there's different directories, right?
Yeah.
but there should be the raw weight.
I don't know where the key. Keyboard
Wait,
is
is it,
different.
oh, it's the other data set, no?
Oh yeah, this is the old data set also.
Oh, yeah.
Maybe can you go to
What's the-
can you go to GitHub?
It's 8.7. I didn't know I was doing this part.
Nice.
I like doing stuff on the fly, right?
And just control click on it.
That's what I try to do with the button.
Okay, there should be somewhere data. This one, yeah.
Yeah.
I think it's already linked. Yeah.
So in there. So the raw should be what they download. Because that's what I converted and uploaded to.
Yeah.
And then
Yeah. And then there's example and then group, I think.
example into one.
Where's the /?
Ah. Oh. I don't know.
Yeah, okay.
Yeah.
So maybe just tell them that. Yeah. Also make the
Distinction between the,
CCMS peaks and
yeah,
raw.
yeah. So if I could have people's attention, something to facilitate you downloading, in terms of if you're using a hotspot or so that you don't have to download the entire data set. The structure of the massive account. So this looks a little bit different. So this is the browser window that's available at massiveucsd.edu. But just to point out that there's different folders that are available in the massive ID. So if you're doing the file browser, the file explorer option, you'll see different folder directories in there. What we really need for tomorrow's activity is just the raw data. So it should be present in that folder under raw. And then each group will have a different set of files. So here you can see, it's kind of hard, but there's the example, which is the instructor led one. But if you if you look for, like, group 1 has their own set of files, group 2 has their own set of files, so instead of downloading 20 gigs, it's like 2.5 or 3 gigs of data, so. And to just draw the distinction between the raw data is still converted to the open file format. It's just different than the CCMS peak values, which are internally converted by the massive server. So again, once you make the connection to massive and find the data, all you need to do is go to raw and then download your group that you signed up for on that sheet. So group 1 or group 2.
How much data is it approximately in total? Is it worth making a personal hotspot, or is it still gonna drain my whole bundle for the next 3 months?
Oh, yeah.
So you.
Very worth. We won't need it.
Okay.
I'll just try to download it and when I'm on the Wi-Fi.
Something that's cheap Wi-Fi.
Yeah, too many guests.
Do you maybe?
We can't get it on that computer either. It's a security thing. That's why I was like, have Daniel do it, so it's like. Okay. Yeah.
Hi, my name is Peter. Serum. Where from? From the UK. From the UK.
Yeah.
I was at the University of Bristol. I'm actually. Oh, really? Oh, you came here to the Serum Institute as well, or to the University? Okay. So you just finished your PhD like a few months ago? I had like stuff. Same place. I already had a close up of 1 year in between.
Like
they were quite deep into the so
I guess we could, I put them down a little bit, but.
It's I'm just starting out on some very targeted stuff. So which could essentially duration and software. Which works really nicely.
Yes.
And I really wanted to see nicely and other things that like living.
Yeah. There's a structure. Yeah.
I group creates and targeted work. So then I would see my game in data analysis because I think manual inspiration is not going to come anymore.
Yeah, if you want to get it.
The PhD is on a few years, so
There's so many like pipelines and things, so you can find like a somewhere.
Also to really know how it's done. The considerations that go into like designing your study and everything. But it's a high book that I wouldn't know what it's like.
Whereabouts do you
Tuberculosis. Okay. Yeah. So I'm trying to develop a new method that applies targeted with proteomics for like large gene mutation on genes with no function. In the hopes of finding new drug paradigm, essentially.
Okay, cool. So how does that work practically? So you have growing like there's a strain. Yeah, and then you can do like counting genes or trying for.
Actually, that's a new PhD that's starting now in September. It's actually going to be that with like this huge critical library. He's gonna like add like provide and see what genes are like conditionally essential. And then you know you should see like with this CRISPR